in horses where we could predict their color. So another kind of information that you could actually gain on is identifying those people that are related. And when you have two skeletons, you don't know if they are dad and son or mom and dad or not related. You only have the skeleton. Right? It's hard to tell who is my dad based on my bones, I guess. So if you have the genomes yet, you can tell whether they are too close to not be related. Right? So this has been illustrated by a study focusing on Neanderthals and specimens that were excavated not far from here in the Altai, in the southeastern Russia, north of Mongolia. And there are a number of Neanderthal individuals that were excavated there. So what they have done first, and this is the name of the samples, it's probably close to 25 ancient Neanderthal, 12 and 2 Neanderthals that have been sequenced. And this is the coverage of the genome that they managed. So for a decent fraction, probably 14 indeed, they realized a one-fold genome, or even up to a 50-fold coverage genome from the data. So they could actually generate good quality data. So if you look at the mitochondrial DNA or the Y chromosome, you realize that the mitochondrial DNA of the ones from Shagirskaya, one of the two caves, are actually very closely related, right? You see that? And whereas the orange sequences from Okladnikov are two different sequences, right? And the Y chromosome, the other ones are females, so they couldn't do that for the orange. But the Y chromosome of the Shagirskaya individual, the four males, are also closely related. So it looks like it could be a family on the maternal side and maybe a family on the paternal side as well, because their mitochondrial DNA and Y chromosomal DNA are closely related. But they did more actually to dive into this hypothesis, is that you could actually calculate the diversity of mitochondrial DNA to the left or Y chromosome to the right, that will be the two bars, in each of the caves that they have identified. So Shagirskaya, and you could compare to other types of data sets that have been generated, for example, in Western Russia here, or Bashokiro, which is in Romania at about the same time. And you realize that the amount of genetic diversity is very low in Shagirskaya. It's similar to what was available at the same time in Western Russia for sapiens populations, but it's a lot less than in Romania, right? So what you learn here is that probably both for the Y and for the mitochondrial DNA, the population was not really diverse, meaning that the population size of the population was fairly limited, very small. Otherwise, there will be more diversity. You remember that yesterday we said the for NE, the effective size is related to the time it takes for alleles to reach fixation by genetic drift. Right? So if populations are small, the genetic diversity will not maintain too long. This is an illustration of this principle. And what they do even more is that this is the map of the different samples they identify in the Shagirskaya cave, is that you find bones split apart in the fossil record. They could identify that some bones were actually from the same individual. Right? So that bone that was found here and that bone that was found here was actually the same individual. And when you find bones, you never know if it's from the same individual or not. So you realize how the enrichment that such data gives you, because it gives you a better understanding of how many individuals were actually represented, how they look like, and even if they were males or females or living in large or small populations from those. Okay? So we can go even further, and actually with the DNA, you could do also population analysis like we saw yesterday. And so here I'm going to illustrate the principle with two examples where you could actually learn something about the migration of the people in the past. And I'm going to illustrate that for us with the first example, the first Paleo-Eskimo genome that we sequenced in 2010. As I told you, it was an Eskimo living in southwest Greenland 4,000 years ago. And by the way, the sample we had was hair. Remember what I've told you? Hair is good. Right? It's rich in material. Okay, so we did that. And when we did that, we were able to characterize at a very high coverage the genome at 20x coverage, so 20-fold every single position, and so that revealed close to 300,000 genotypes of that individual. So what we decided to do is place this individual in the map of the worldwide human diversity. So each of the points or triangles that you see is actually a modern individual living today in Eastern Asia, Europe, or in America. There are thousands of such individuals in the plot. And what we show with the star is just where our ancient individual projects is placed, if you want, okay? And so the reason why we wanted to do that is to just ask a very simple question. Is an Eskimo living in Greenland 4,000 years ago closer to the people that live in Greenland today? That could have been a fair explanation, right? You would expect that maybe from the same place, which is fairly remote, fairly hostile, because it's a hard environment to actually survive in, maybe people who have reached that place, who have only succeeded in reaching that place once. So our expectation would have been that they were the same. And in fact, they were not, because look at where the ancient individual is, but look at where the modern Eskimo people living in Greenland are located. Okay. This is all the triangles. And there are people in the world today, living today, that are closer to the ancient Eskimo than the modern people from Greenland are. And those people are not randomly. They are the Chukchuk, the Koryak, and the Yukagirs, which are the people living in northeast Siberia today. In Siberia. So what you learn is that probably the history of the peopling of Greenland is not a single migration of people discovering Greenland 4,000 years ago and living in Greenland until today. It's a wrong model, because those from today are not the same genetically as those from 4,000 years ago. So you have to realize that there has been a first migration succeeding in peopling the Greenland, but this first migration was replaced by a second migration, and the second migration, we could prove in the follow-up study 5 years later, created the modern peoples that live in Greenland today, and this is the so-called Inuit migration that took place about 800 years to 1,000 years ago. Okay? So what you learn here is that the peopling of Greenland was more complex than what we would have expected. So basically you unveil the real complexity of the past migration history of the world. And since this first paper, the game in ancient DNA has mostly been that one, to try to reconstruct the history of migrations of people and animals in the past. So I'm going to illustrate another very important example of finding that has been done, in this case not in Greenland anymore, but in Western Europe. So this is Western Europe, you have France here, you have Anatolia here, and here you have what we call the Western Steppe, if you will, right? And so we're going to try to understand how the people have inhabited that place over the time. So one methodology that is always used in ancient DNA studies is the principal component analysis that we covered yesterday. So in that space, the gray triangle will show the modern diversity of people living in Western Europe today. Okay? Every gray point is someone living today in this part of the world. Okay? And what you do here is you project the placement of ancient individual in this diversity. Okay, so if we take ancient people who lived in the Neolithic in Turkey, in Anatolia, they will project it. If you take people who lived in the steppe 5,000 years ago, they will project it. And if you take some people living in before the Neolithic period, in the Upper Paleolithic or Mesolithic of, say, France, they will be, okay? That's just to give you a placement. So what you learn is that the people that used to live in France in the past, say 20,000 years ago, are not the same genetically as those that live today in France. So you realize that in the last 20,000 years, people have come and went. So there has been movements of population. So let's try now to focus on UK. And let's take some people who lived in UK in the Mesolithic, before the Neolithic Revolution, before we became farmers. So this is where they project. So it tells you that people who lived before farming in the UK, they are like the people that lived in France before the Neolithic. Okay, they are typical of that first population. What happens when people come and become farmers? Well, they project it. They project not with the local people of the past, but somehow intermediate between the local people and those who invented farming in Turkey. So it means probably that some people left Turkey, reached out to UK, and mixed together with the local population. And in fact, because they are closer to the farmers than they are to the local people, you could probably say, by and large, that the majority of the genome comes from the farmers. So what you learn is that there's been new people coming from far, actually coming in UK, and mixing with the local people in a way in which there were more contributions of farmers. And from now on, this is the people living in UK from the Neolithic. Let's go one step further. Let's have a look at the people living in the UK in the Bronze Age, which is, say, 4,000 years ago, 3,500 years ago in UK. This is where they project. And again, you see that this is not anymore any of the two previous. This is somewhat intermediate between people living in the steppe 5,000 years ago and people who lived in UK after they came from Turkey, right? So clearly there's been another mixing. Maybe this time is half and half because it's half the way between those two groups. That tells you that another group of people came into UK and mixed with the local population that was itself mixed with the local people, because the Turkish people who have actually mixed. Okay? So you see how simple information about the past can actually slice the past in a way so that you could see the movements of people as it unfolded during history. But it's not that all, because in work that we have published a few years ago, we have identified some farmers from France who are actually closer to the pre-farming than actually to the farmers. So it's not a wave that was massive and necessarily replaced everyone. It's actually depending on the population. Some local groups remain more non-farmers, so hunter-gatherers, than the farmers coming from Anatolia. So that's one thing that you can do. You can also do more complex things like retracing the population histories, in that case of Neanderthals. What I mean by that is that we know a lot about Neanderthals. For example, we know their skulls, and we know that this one is a Neanderthal, because if you compare this skull to our skull, you don't have any chin here. You see no chin. You have a large back part of the skull, and you have here suborbital parts of the skull that are very pronounced in them that we don't have, okay? So we know they are anatomically different from us, and that's how we actually have discovered them. They don't have the same anatomy, whereas we have a modern anatomy. That's how we make a difference. And the Neanderthals, they were living in Europe and up to the Altai from 300,000 years ago to about 40,000 years ago, when they became extinct. And since we have discovered them, the question of whether or not we have got kids with them has been a puzzle to paleoanthropologists, okay? And the reason why anthropologists could not answer this question is that they had a sort of ambiguous fossil record. You have, for example, that skull that was discovered in Romania 40,000 years ago, and that skull actually has Neanderthal features. The back is pronounced, but lacks some other Neanderthal features. And so sometimes our anthropologists have said, maybe this is a hybrid between Neanderthals and us, our ancestors. And so maybe they could have a kid, because we may have found a hybrid. But it's hard to believe that this is a hybrid necessarily. It could be a badly preserved skull, it could be just a weird anatomy for that individual. So how about the frequency of mixture between Neanderthals and us, our ancestors? And this this question has been solved almost 15 years ago by the sequencing of the Neanderthal genome, developing this sort of D-statistics. So I'm going to tell you something about this test, because it's quite a simple and powerful test to test for a mixture. So imagine that you have 4 genomes. The genome of an outgroup, this is the chimpanzee in that case, but we could have taken any other outgroup. Imagine you have the Neanderthal genome. And now what you're doing is you compare 2 types of genome. one from a Eurasian individual, say a European individual, and one from an African individual. The reason why you do that is that I told you that Neanderthals were only found in Eurasia. You remember, from the Altai to France and Portugal, in fact. So if we have got kids with Neanderthals, you will expect that those kids would have been living and surviving more in Europe than where they were not present than in Africa, right? So your expectation is that you will have an excess of gene variation between Europe and Neanderthals relative to Africa, basically. And this is precisely what the D-test does to test the balance of allele sharing between populations. So imagine that you will look at particular sites across the genomes. Some sites, instead of using C, A, C, T, the 4 letters of DNA, I use A and B, right? A means ancestral, it means the exact allele you find in the output, in that case the chimpanzee, right? And B will mean derived mutation. So if the C is the ancestral, maybe the G is the derived, right? It's not, it's a mutation. So you will look for such instances in the genome where the African genome shares the derived allele with the Neanderthal, right? It means those two, they share affinities because they have a non-ancestral mutation. It's common to both, right? So maybe they have exchanged that particular value. And you will do the same for the reciprocal test. Places in the genome where Eurasian, Europeans share more with Neanderthal than they do with African, okay? So if you look at those particular ABA and BABA sites, ABA and BABA, the D is just ABA minus BABA, or BABA minus ABA. That's the difference between both, right? And this is the expectation you have that if there is no gene flow between Neanderthal and Europeans, there should be an equal proportion of ABA and BABA sites, because there will be no gene flow that bring B mutation into Europeans. So there will be no excess of BABA sites, okay? So the test really tests for that. Is there equal amount statistically of ABA and BABA sites in the genome in 4 ways alignment? And if you find it's the same, it means no gene flow. But if you find this, it means gene flow with Africa. If you find an excess of BABA, it means excess of shared alleles with Europeans. What is it they found? Exactly that. So they found that there is an excess of shared derived alleles between modern Europeans and Neanderthals, which means that not only our ancestors in Europe and Eurasia had kids with Neanderthals, but those kids had kids themselves that had a legacy up to the modern populations. The genes that were transferred from Neanderthal to us were transferred to us up until today. And this is about 2% of the genome that we have across Eurasia that we have from Neanderthal ancestors. You have a bit more in Asia for very complex reasons. It's more closer to 3%. And in Africa, they have a lack of it. There's only in Northern Africa, in the north, that you have a bit Neanderthal because it's people leaving Africa, mixing with Neanderthals and coming back in Africa, right? So it's a bit more complex than what I told you. So you learn that. You learn even the legacy of populations that are extinct now and yet have met our ancestors more than 40,000 years ago. In fact, it will also date the time of admixture, and we date it to about 55,000 years. We can learn also about populations we didn't even know they existed, such as the Denisova. So the Denisova cave is found here in the Altai. This is the entrance of the cave. If you go into the cave, you will find people excavating, probably now, in this time of the year. And they have been discovering that bone, as I've told you, right? It's a pink bone, it's small, but you don't know what that thing is. In fact, when they discovered, they didn't even know it would be a human, right? Because it looks like anything. And you will realize that when you find that, it's hard to know what it is. So it's hard to make a theory about anything. So probably these findings in classical archaeology without DNA will be sort of lost. It will not give any information, basically, except that there is a bone in that cave. Yet they did DNA extraction, and what they discovered is that if you focus on the genome, the nuclear genome here, this individual will place in the tree as a sister group to what is shown in orange, which is Neanderthal. And it's closer to Neanderthals than it is to us, anatomically modern humans that I show in blue. But it's not a Neanderthal, because look at the distance you have from Neanderthals to the Denisovan. So what you learn is that it's a population of hominins that is now extinct, that doesn't belong to us today, but that was closer in terms of proximity of populations to Neanderthals than they were to us. Yet they were very different from Neanderthals, right? So you learn that there was a third group of humans that we had no idea even existed before the pinky bone was sequenced, right? So you see the power from a bone that wouldn't have told anything. You learn that this is a group of hominids that was living in Asia at some point in the past. And because you could do the test, the Ababadala test, with any modern populations, they have been able to show that these Denisovan have contributed an excess of allelic ancestries to Papuans, people living in Papua New Guinea today, somewhere else in Asia, right? So that means that probably, even though only one individual was sequenced at the time in the Altai, probably the Denisovan range was larger than just the Altai, because they have now people descending from them or having contribution from them that live somewhere in Indonesia or Papua New Guinea. Right? And actually, because of these types of findings, now we know the human tree is more complex than what we thought it was. There is us, there is Neanderthals, but there is a third player. And what the work has revealed is that those groups exchange genes. They mate many times, in fact, in different parts of the world. So we have a very more mixed history of human evolution than we thought before. So I focused a lot on humans not because humans are necessarily more interesting. It's simply because 90% of what people do in ancient DNA is human DNA. But my group does a lot of animals. So since the lecture is about animal domestication, I thought that for the 15 minutes that I have left or something like that, 15 minutes that I have left, I will focus a bit more on the animals. And I'm gonna go to horses to show you why we have been interested in sequencing many genomes from horses that lived in the past as opposed to just humans. And there are two reasons for that, really. Is that if you have ancient DNA data, where today will be here on the x-axis, 0, and the past will be in negative values, somewhere in the past, if you have DNA data, you may be able to reconstruct allelic frequencies through time. And if you have a trajectory of an allele that didn't exist, start to emerge, and then goes up in frequency, it may be the sign of selection. It may be the sign that this allele became more and more popular through time. Maybe because some people selected the carriers of that allele. It's not a demonstration, but it's a good sign it has been the case. So if you have ancient DNA data, maybe you will be able to identify which parts of the genomes look like that. And those places in the genome are good candidates for mutations that we have selected in the past to create the animals that we like most. First reason, we may understand better the selection process by which people have created domesticated animals. You remember yesterday I mentioned the TBX3 example that people here, Lin and Husher, have been publishing in horses from Asia, where a TBX3 variant, a G variation on the TBX3 gene, is actually responsible for 20% of the size variation of horses in Asia. So one A to G change in this gene location changes the size of the animal big time, right? So we were interested in looking at the allele frequency of that through time, and so we generated a few years ago a very large panel of horses from the past, more than a few hundreds of ancient genomes through time. Because we had such a panel, we were able to reconstruct the allelic frequency of the G allele through time. And I'm sorry, I switched the x-axis, so now today is here, and the past is to your right. And you see that it started to increase about 2.3, 2.5 thousand years ago, which is almost coincidental with the rise of the Qin dynasty in China. And so we think that Qin, one of the first things they have done is actually selecting for larger horses. Because the Qin, they needed to have an empire that was homogeneous, and they elaborated modern China, as you know better than I do, and part of the success of their federation of states was to have a powerful military, basically, and having larger horses was key in this particular context, especially as you had the nomads from Mongolia attacking China. We could go just beyond the DNA of the horse or the animals or the humans that you are interested in. You could also learn about whether they were infected by pathogens. So one pathogen that I'm going to take as an example is this nasty bacteria that we call Yersinia pestis, and that is responsible for plague disease that still today can kill people in countries such as Peru, Congo, or Madagascar, for example. And in the past, plague has really killed a lot of people. For example, in the 14th century, in Europe, plague has killed half of the human population, because there's been the Black Death pandemics that came and killed half the population. Okay? So plague has been really a bugger in the recent history of human populations. How do you get plague today? There are two ways. You could get someone who has plague and coughs, and by coughing, they will actually infect you. When they cough, you breathe the particles that are emitted by the person, so that means that you get infected in your lungs. It's a respiratory disease, and if you develop the respiratory disease, well, this is a bad case for you because you're going to die. Right? If you're not treated by antibiotics, you're going to die. But there is another way to get the plague, is that some animals are infected, and some lice that will actually bite the animals because they want their blood, will actually suck the bacteria, and those lice, they will be even contaminated. I said lice, I'm sorry, it's a flea. I'm sorry, I don't know why I say lice, it's a flea, sorry. It's not the same kind of parasite. So it's a flea. And the flea will be infected in this gut because it swallows blood with the bacteria. And the flea can jump from an animal to you and will bite you to get your blood. And by getting your blood, will actually infect you. Right? And so the process by which, by the way, the flea will contaminate you, will infect you, is nasty, because actually the bacteria is adapted to actually block the digestion of the flea. So the flea gets more blood, and so much blood that they can't actually swallow the blood anymore, so they get filled up to they reject the blood back in your body. And this is this process of them rejecting your own blood in your body. As your blood goes in their body and goes back in you, it gets contaminated by the bacteria, basically. The bacteria create a biofilm in the stomach of the flea, and this biofilm blocks the stomach so much so that then the blood has to go back to your own body. Right? So there is an adaptation of the bacteria that is very well known at the molecular level. So there are the two ways. And we didn't know when plague started to infect our populations. I told you that in the 14th century we know of the Black Death. This is called the Southern Plague pandemics in the 14th century. And these pandemics lasted up until the early 19th century, in fact, killing half of the European population. We knew from historical record that there was another plague pandemic, the first. It was during the 6th to the 8th century, and some historians say that the first plague actually caused the collapse of the Roman Empire, because it also killed a lot of people in the Roman Empire. But since it was called the first, it means that historians have no trace of plague epidemics before the first pandemic. So we thought, because of historical sources not being complete, that no plague was actually a problem for human populations before the 6th century of our era. So maybe plague was a problem for us for 1,000 years, 1,500 years. But actually, some people have sequenced humans from this part of the world that date back to the Neolithic, the end of the Neolithic, 4.8 thousand years. And what is it they found? They found the DNA of plague in the human body. So all of a sudden, doing DNA, even if the bones are the same, if you die from plague or not, you realize that some people had the plague almost 5,000 years ago, way before we thought even plague existed. And in fact, because you have a genome, you could do a number of genomic comparisons we learned doing over the last few days. So the genome of a bacteria such as Yersinia pestis is one circular chromosome, that's a bacteria, no nucleus, and there is 3 plasmids that you see here. So what I show here is the coverage of the chromosome and each plasmid for different types of plagues that have been characterized. Right? So you see that the modern plague, they all have the whole chromosome, they all have the plasmid, and the plasmid, right? And what you look in the different colors is the coverage you have in ancient DNA data from the Black Death, from the first epidemics, the Justinian at the end of the Roman Empire, or the Bronze Age, late Neolithic that I show in blue. And why this is interesting is that if you look closely, you will find regions that are totally uninteresting, like here. There is nothing, everything is covered, right? But if you look closely to places like that, you realize that the modern plague have that part of the plasmid, the first and second pandemics as well, but there is no data for the blue, where there is data there. So it means that there was a deletion here, in the ancient plagues from the late Neolithic. And it happens that there is a very important gene there. This gene is called YMT, and in this process of biofilm formation that I've mentioned, this gene is key. So what you learn here is that because there was that gene that was lacking in the late Neolithic plague, it probably means that the plague from the time in the past, 5,000 years ago, could not make the biofilm on the fleas, so probably that the plague could not be transmitted by the fleas. So it could only be transmitted by someone coughing, probably. So you see that by molecular evidence, you even learn about ways pathogens will be infecting populations in the past, right? And even if you have genomes, you could not just look at presence absence of gene, but you could actually try to find mutations that would be adaptive, as we saw this morning with the Marek disease, for example. And what people have confirmed is that indeed, this is not just the YMT gene that emerged at that point in time, that's a number of mutations that are key to make the biofilm. So it's the whole toolkit of a bacteria that makes them able to do the biofilm that emerged somewhere around 3.5 thousand years ago, and that enabled the plague to find a new vector, the flea, to infect a larger diversity of mammals, simply because they exploit another parasite to actually get to their So since I showed a pathogen, and since I told you that this morning that this is very important to understand microbiomes, I'm going to try to show you 2 things now, and I'm going to try to be very quick, because Juju is standing up now. The first thing that I'm going to